Hi, so far I have not shared any videos related to NEET exam content in this channel. So this is the first attempt. Mr. Karthikeyan, a biology faculty in Chennai, has volunteered to do this video for this channel. He has spoken about the topic biotechnology principles. I hope you like it. If you have any specific comments about this video or this topic, you can leave a comment. If you want this channel to cover any particular topic or you have any particular question about NEET exam or NEET exam content, please post the question. I will ask Mr. Karthikeyan to cover those questions in the upcoming videos. I hope this is useful. If you like this video, please click the like button. And if you have not subscribed yet, please subscribe to this channel and click the bell icon so that you don't miss any important video from this channel. Now let's go ahead and watch what Karthikeyan has to share about the biotechnology principles.
So let's get started with biotechnology principles. So we'll be looking on certain important NEET aspects that could be asked in your MCQ examination. So I will not be focusing on the entire chapter, rather I'll be focusing on certain important facts that could be asked in the exam, some important conceptual understanding that is required. Just for the namesake, we'll just start with the introduction, that is the definition of the biotechnology that is given by a federation called EFP. This had been asked once or twice in MCQs, asking for abbreviation of EFP. So EFP stands for European Federation of Biotechnology, that defines biotechnology as the integration of natural sciences and organisms, cells, parts thereof, and molecular analogs for products and services. So this definition says, for a biotechnological approach, you require an organisms, cells, and also the molecular analogs. What are the enzymes, etc., that is required for undergoing various biotechnological processes. So only if you bring all these components together, a biotechnological process is possible. So EFP states the integration of all these components is essential. So from this slide, the carryover point is abbreviation of EFB and the terminology called integration. Very, very important. Now, this slide is of utmost importance among all the slides that I'm going to tell, that is findings. So in your NEET question paper, you will have at least 4 or 5 questions that are going to come from these scientists' findings, their year of discovery and what they discovered. So from this chapter, the first and foremost important finding that you must understand here is about the recombinant DNA. So what is this recombinant DNA? So the word itself is self-explanatory. So recombinant means you're combining. So you have got some particular piece, you have introduced newly, and you have got a new combination. Like what you say in genetics, recombination of 2 parent genes is going to give rise to a new offspring. So that is called as recombination. So when 2 entities are going to combine, it is called as a recombinant DNA. So the first recombinant DNA was constructed by Stanley Cohen and Herbert Boyer in the year 1972. The year of discovery is the most important thing among all the discoveries. So Stanley Cohen and Herbert Boyer, they discovered the first recombinant DNA by isolating antibiotic resistance genes. And the next important finding is father of genetic engineering is none other than Paul Berg. Again, very, very, very important scientist. This was asked twice in the ENTS examination. So what he actually did, why he was termed as father of genetic engineering, is he worked with bacteriophage. So what do you understand when I say bacteriophage? So phage means killing, it's bacteria. So there are certain cells, or bacteriophage is nothing but viruses that kill bacteria are called bacteriophage. So what he did was, with the help of a bacteriophage called lambda phage, he was able to transfer gene of SV40. What is this SV40 is? It is a strain of a virus group called simian virus, SIMIAN, simian virus. So what he did is he was able to transfer this gene of SV40 variety into E. coli with the help of lactophage. So since he was able to show the working of a bacteriophage, he was called as the father of genetic engineering. So this page is very, very important. Who constructed the first recombinant DNA in which year? And then Paul Berg. These two entities are very, very, very important. And the third important thing that you must know is about plasmid. So what is a plasmid? As such, you don't have any scientist name, but this plasmid is very, very important. Let's talk about biotechnology. So what is a plasmid? Autonomously replicating, which means it replicates independently. Autonomous means independent. So autonomously replicating extra chromosomal circular DNA in bacterial cell. I'll repeat, autonomously replicating extra chromosomal circular DNA in the bacterial cell is called as a plasmid. So this is mostly used as a vector because we say extra chromosomal. So there's going to be an additional chromosomal entity which is going to be autonomously replicating in bacterial cells. So that is called as a plasmid, which is mostly used as a vector. So you must understand this. Now, principles of biotechnology, again, just for inclusion's sake, I have included here. So you have two principles, one is genetic engineering and maintenance of sterile ambience in various processes. Okay, so genetic engineering means you are going to use all the chemicals and other chemical entities such as your enzymes, all culture medium in which you're going to grow these cells. So all these you're going to use in order to get a particular output. That is genetic engineering. And when during the course of the entire process that you're going to carry forward, you require sterile conditions. If you're not going to provide sterile conditions, then that could be an incomplete biotechnological process, or you will not get the proper output. So these two are crucial principles, or you can call it as a crucial events that is going to happen in biotechnology. Now, first let us start with gene cloning. So usually when you take, you will in the NEET, you will have 2 or 3 questions asking you to arrange the steps in order. So arrange the following gene cloning steps in order. Arrange the following gene modifying organisms in order. So you will have definitely 2 or 3 questions will arrange the steps in order, something like that. So for that I have included this. So always understand. So let me make you understand this concept by giving the present situation. It's an hypothetical situation. Probably we can expect this to come in few more weeks or days or months or years, whatever. So let us take this COVID-19. Say suppose there's a scientist in some part of the world. He has studied the virus and say suppose he has identified the antigenic structure. So he has told, okay, this is the antigen that is responsible for the virulence property of the virus. And he has also designed a gene which can destroy this virulence property. That is, which is going to be an antibiotic effect to this particular antigen. Okay, so he has designed a gene, so he has identified an antigenic structure of virus. He has said this is responsible for its virulence. And also he has said, suppose he has identified a gene or he has designed a gene that can possibly be an antibiotic action on this particular antigen. So at the present situation, if such condition exists, so what will the scientists do? They will just simply take this gene, they will introduce into the patients, and subsequently will find the effects. So in process of a gene cloning or in process of genetically modified organisms, the first and foremost step will be identification of a foreign piece of a DNA or identification of gene of interest. I suppose I work on gene A, suppose you work on gene B. So you need to identify your own gene of interest in which you're supposed to work. So the first and foremost step will be that is going to be identification of foreign piece of DNA or gene of interest. So that's why here we have got this foreign DNA, meaning all the desired gene he has given. So first step is going to be identification. And simultaneously you are going to have a vector. So as I said, plasmid is the most commonest vector used in almost all biotechnological processes. So what is going to happen will be, we'll be discussing about the enzymes regarding this restriction digestion. What is going to happen? There are going to be certain enzymes which is going to identify the sequences which are common for both your plasmid and also in your foreign piece of DNA. Say suppose plasmid is made up of 1000 sequences. The foreign piece of DNA is made up of 500 sequences. In both these sequences, there will definitely be a part of the DNA where the sequences are going to be similar or the same in both. Which means, say suppose here, the blue region is going to correspond to your this foreign piece of DNA. So this blue region is same as that of this red region. So the enzyme is going to make cuts at the same site or at the same set of sequences at both this blue region as well as this red region. I will understand. So we'll study in detail. So just try to understand there's going to be a restriction enzyme which is going to make cuts at same sequences of both this blue region. So that's going to be identification, and then you're going to be something called ligation. So this process is nothing but introduction of your foreign piece of DNA or the gene of interest into your host. So through the vector, so this vector is going to be taken into your host, that is your E. coli cell. So where there is going to be multiplication of this desired piece of cells. So for the multiplication of this desired piece of cell, you require maintenance of sterile conditions. Okay, so this is the picture that is going to depict you the cloning processes. So you are transported into the host cell. There's going to be multiplication of this rDNA molecule, and there's going to be division, and subsequently there's going to be clone forms. What is clone? That is, you're going to get thousands of cells of one particular type. That is called as a clone. Okay, so I want this particular piece of cell to be copied in thousands of cells. So this is gene cloning. So these are the three basics. I hope you understood. Okay, so we'll be learning about the restriction, ligation, and all shortly. So as I said, the three basic steps in gene cloning are genetically modified organisms. You identify the DNA of desirable genes or gene of interest. You introduce this particular identified DNA into the host, and you are going to maintain this introduced DNA in the host and transfer of DNA into its progeny. So for all these two steps to happen, that is the maintenance and then transfer of DNA into progeny, you require proper sterile conditions to be maintained. Now, we'll move on to tools of recombinant DNA technology. So, this tools of recombinant DNA technology is nothing but what I explained about this restricted digestion environment. So, as I said in the initial, we saw EFP different as integration of your organisms, cells, the molecular analogs. So, all these, so all your chemical constituents, main of what we're going to use is going to be your restriction enzymes. So what are these restriction enzymes? These restriction enzymes are a group of enzymes that is going to belong to a class called nucleases. So you know there are various types of enzymes. Hope you would have studied the biomolecules. These restriction enzymes are going to belong to a class called as nucleases. So what is the use? What are the types of restriction enzymes you have? Is you are going to have two types. One is an exon nuclease, other one is an endonuclease. This is a very important point. What is the basic difference or the major difference between an exonuclease and the endonucleases? If you take exonucleases, these enzymes remove the nucleotides from the ends of the DNA at the extreme ends, either at your N terminal or C terminal, like how you find an amino acid. Here in DNA, you will have 3' or 5'. So at the ends of the DNA molecule, if it is going to cut the piece of DNA, it is called exonuclease. Exo means outside. So exonuclease, ends of the DNA molecule. Whereas if I'm going to take about this endonucleases, endo means inside. So the endo, it's not, it is not an apt word here to use inside. But what I can say is they are going to make cuts at specific position with the DNA. If you observe this gene cloning slide page, here we have put this restriction site. Right, so this blue color is restriction site. I told this enzyme is going to recognize a same set of sequence in both your vector DNA as well as in your foreign DNA. Right. So now this endonuclease, if you take, I will show you a picture. Okay, I'll show it later. Okay, so this endonuclease, if you take, it is going to recognize a specific sequence called palindromic nucleotide sequence. sequences. So you must be knowing this palindromic nucleotide sequences. What are these palindromic nucleotide sequences? Palindromic nucleotide sequences, that is from forwards if you read a particular sequence, or from the backwards, it is from either left to right or right to left if you're going to read, the sequence is going to be the same. So GAATCs, I will give you an example. So palindromic sequence of the sequences, which when read from left to right or right to left, is going to mean the same. Okay. So what are these restriction enzymes? This is a very, very important slide. You will get definitely one MCQ from this particular slide. So restriction enzymes are obtained from your bacteria. On that important point, the next 2 points are important. Again, you have a scientist discovery here. So the second important is very, very, very important point. It brings about fragmentation of viral DNA without affecting bacterial genome. So what it's trying to tell is, it is protecting the bacterial DNA from the foreign DNA. So mostly in your NCERT textbooks, you will not find this point. You would have simply given molecular scissors, or it is used to make cuts at specific ends or at the ends of the DNA. So restriction enzymes. But this point has been asked in MCQ very recently in 2017. Also in JIPMA they have asked, in NEET they have asked. So this is a very, very, very important point. Restriction enzymes, it confers protection of bacterial DNA from the foreign DNA. The very, very, very important point. So it's going to bring about fragmentation of viral DNA without affecting your bacterial genome. Hence, we are trying to say it is going to protect bacterial DNA from the foreign DNA. And the third important point, so the first discovered a restriction enzyme was EcoR1, which was discovered by 3 scientists, Arbor, Smith, and Nathans, in the year 1978. Now, we'll just see working of the restriction enzymes, how it actually works. Very, very first and foremost important point. So they may ask, sequence the steps how a restriction enzyme is going to act. It is also a very important question. So the first and foremost step that is going to happen in the working of restriction enzymes is going to be inspecting the length of DNA. So we saw in gene cloning picture, there was a blue region in the vector, that is the plasmid, and there was a red region in the desired gene. And I said these blue region and the red region is going to match each other, right? So what will this enzyme do is, it will start by inspecting the entire length of DNA. It's an NCERT line again, inspects the entire length of the DNA. Inspecting, it is going to check whether I'm getting this particular sequence or not. So every restriction enzyme has got its own palindromic sequences. I told palindromic sequences, if you're going to read from left to right or right to left, it is going to mean the same. So if you take this particular example here you have GAATTC. So I have read from left to right in the upper strand. If I read from right to left in the lower strand, it is going to mean the same. So like your madam, MADAM. There I read from left to right or from right to left, it is going to spell the same. So same way is called as a palindromic sequence. So these restriction enzymes are going to identify or work by inspecting the entire length of the DNA molecule. So it has inspected all over the DNA molecule and it has found this restriction or this palindromic sequences. Once it has found this palindromic sequences, it is going to make cut at the same positions, meaning in the vector DNA as well as in the foreign DNA, is going to make a cut between G and A only. So if you take in the upper strand of the sequence, it is making cut between G and A. And in the lower strand of the vector DNA, it is making cut at G and A. And same thing is going to happen here also. So it is going to make cut at the same sides of the opposite strands. This is a very, very, very important carry forward point. Okay, so now what is going to happen? So it has inspected the length, it has found the palindromic sequences that may cut at the same site of your DNA molecule of opposite strands. Once it has made such a particular thing, there is going to be production of sticky ends. Very, very important point. What is the advantage of sticky ends production? Once there is going to be sticky end production, these sticky ends is going to facilitate the combination of these both strands easily. So bond formation between the vector DNA and your foreign DNA is going to be easier. Or you can say action of DNA ligase is going to be easier when you have sticky ends of the same type. Very, very important. Sticky ends facilitate the ligation of your DNA strands. As simple as that. And then you're going to have formation of recombinant DNA. So what you must understand, inspecting of length of DNA molecule, identification of palindromic sequences, makes cut at same sites of opposite strands of DNA. And then there is going to be production of sticky ends, and there is going to be ligation or joining of sticky ends. What is the use of sticky ends? Sticky ends are going to facilitate the formation of the bonds between the two strands. This is the understanding that you must need. Okay. Now moving on to vectors. So vectors, if you take any vector, as should have these three particular components. One is your ORI, stands for origin of replication. Okay, what is the use is very, very again important MCQ. Sequence is responsible for copy number of the linked DNA. Copy number means what amount of cells that you're going to get is nothing but a copy number. So the origin of replication, it is going to consist of sequence from where replication of the desired gene is going to begin. At the same time, its significance is responsible for the copy number of the linked DNA. So what amount of cells you're going to get is nothing but your copy number. Notice the selectable marker. Genes encoding resistance to antibiotics is going to be a selectable marker. So selectable means you're going to choose. Marker is nothing but any substance that is going to help in a particular phenomenon or a particular observation is nothing but your marker. So selectable marker helps us to identify whether the foreign piece of DNA has actually been incorporated into this. I'll explain you with this cloning sites. So what does a selectable marker means? Genes encoding resistance to antibiotics is selectable marker. Now, and the third component is going to be your cloning sites. So cloning sites means actually where you're going to find the ligation and all that. Okay, so actually where the foreign piece of DNA is going to go and bind is going to be your cloning sites. Okay. So this is the PBR322. It's an example of a vector that is going to be PBR322. If you take this vector consists of two sets of genes. That is your tetracycline resistant genes and ampicillin resistant genes. So if someone asks you what is the selectable marker of PBR322, it is going to be tetracycline resistant genes and ampicillin resistant genes. So the genes which is going to encode for a resistance of a particular antibiotic is nothing but your selectable markers. And you have ORI, origin of replication, responsible for your copy number of a linked DNA. And all these, what you find here, BAM-H1, SAL1, PDU1, PST1, all these are your cloning sites. Now, what is the significance of these cloning sites in a vector? Very, very important. These, take down this point, the cloning sites, if a ligation of a DNA happens at a cloning site, you will be able to appreciate or differentiate transformants from non-transformants. NCERT does not give a clear idea of about it, but I'll explain you. Say suppose I am going to ligate a piece of DNA by means of BAM-H1 restriction enzyme. So if you take BAM-H1 restriction enzyme, right? So this restriction enzyme is going to act at tetracycline resistant gene. So say suppose I have a foreign piece of gene here and I have this vector here. So as I said, a restriction enzyme is going to identify the same sequence in both your vector and then your foreign piece of DNA. Okay, so I have a foreign piece of DNA, so I'm not able to draw, just imagine or listen. So I have a foreign piece of DNA here and I'm going to use BAM-H1. So it is going to identify its BAM-H1 sequence. It is going to make a cut at this particular tetracycline resistant gene. What is going to happen? that new foreign piece of DNA is going to come to this particular site. So we saw the blue region and red region here, right? So if you see here, so see, this particular foreign DNA is being inserted at this particular site because the enzyme is going to identify this portion of your, that is, it is going to identify only this blue region only. Okay, so this is where exactly that restriction enzyme is going to act. Or to be more precise, this is the site where palindromic sequences is going to act. So if you come to this slide, I have a foreign piece of DNA. I use BAM-H1. So it is going to make a cut here. So the foreign piece of DNA is going to be inserted at this particular site. What is going to happen as a result? I have introduced a new piece of DNA at tetracycline resistant region. So this could have lost resistance to tetracycline. This could have lost resistance to tetracycline because there is going to be some genetic alteration taking place here, because the kind of bond that is going to form, all that will vary. There's going to be a new variation or an alteration happening in this particular site. Okay, now how it is going to help me in identifying transformer and non-transformers? So now you should understand the introduction of foreign piece of DNA at this tetracycline resistant site has made this particular organism lose resistance to tetracycline. Again, I'll repeat, you have introduced the foreign piece of DNA into this tetracycline resistant gene, hence the organism or the host will lose resistance to tetracycline. So, this recombinant DNA will have only resistance to ampicillin and not tetracycline. So, what the scientists did, they took two different culture media. On one culture media, they had this ampicillin antibiotics, and on the other culture media, they had tetracycline antibiotics. Okay, so now you just imagine if I introduce recombinant DNA into tetracycline medium, the organism will die because it has lost resistance to tetracycline. But if I introduce a non-recombinant, meaning the same cloning vector which has both resistance, if I introduce into tetracycline medium, it will grow. So this is how you can differentiate recombinants from non-recombinants. But explanation itself is taking so much time, it is complicated. Imagine the procedure. So there was one more new simpler technique that was introduced. Again, a very, very important NEET question, that is insertional inactivation. So what is this insertional inactivation? Again, self-explanatory. Okay, so what they did, they took two culture media, whether recombinant, non-recombinant. To the culture media, they introduced a chromogenic substrate. What is a chromogenic substrate? Is the one that is going to produce color. So substrate that is going to produce color is called as a chromogenic substrate. So the beta galactosidase enzyme is commonly used as a chromogenic substrate. This galactosidase enzyme was used as a chromogenic substrate. How this is going to help? Understand, if you have a normal vector molecule, it is a non-recombinant. Okay, so this non-recombinant is not going to have any insert, meaning there is no foreign DNA that is being inserted into the vector. Since there is no insertion of this foreign piece of DNA into the vector, this beta galactosidase enzyme has a place for insertion. So when you introduce into the media, it is going to produce color. So non-recombinants will produce color if beta galactosidase is introduced. Whereas if recombinants are put in the culture containing beta galactosidase, it will not produce any color because it has got an insert, meaning a foreign DNA. That is why it is called insertion inactivation. It's a very, very important question. How will you differentiate recombinants from non-recombinants by using insertion inactivation? So they give you four options. So production of blue colored colonies in absence of an insert is non-recombinant. Okay, this is the major principle. Presence of or production of blue colored colonies in absence of an insert is the principle underlying. Okay, please try to understand. Now, this is again an important point. What you must know is Agrobacterium tumifaciens is a pathogen of several dicot plants. It delivers tDNA. What is this tDNA? Tumor DNA. So this pathogen, this DNA is going to convert all your normal cells into tumor cells, and it is going to assist, or it is going to advise these tumor cells to produce chemicals that will favor the growth of pathogens. And later, this particular gene was introduced as a vector. So where it is mainly used, it is used in transfer of genes in crop plants. Very, very important point. Which of the following species is being used for transfer of genes in crop plants? 2018 NEET question. It is going to be Agrobacterium tumifaciens. Again, scientist, synthetic DNA. So Kornberg in 1961, he synthesized first synthetic DNA from nature. So all these you study, not of that significance, but Kornberg in 1961 synthesized the first synthetic DNA. Very, very important point. And Corona, okay, should not fear. It's the name of the scientist, Corona. You can pronounce it as Corona. Corona in 1968, he synthesized first artificial gene without a template. Because without any reference, he just synthesized some artificial gene. Actually, what he did was he was able to synthesize a tyrosine related E. coli gene. Okay, so that was the gene that he synthesized. Okay, so tyrosine related E. coli gene. If you want to just note it down, it is of not that importance. So you should know scientists of the era of discovery. Okay. Now, the last important concept, PCR, again scientists, so Carey Mullis 1983, and main specialty is Taq polymerase enzyme was isolated from Thermus aquaticus bacterium. So this enzyme, it is again a type of DNA polymerase enzyme, it was used in PCR, polymerase chain reaction. What is the purpose of doing polymerase chain reaction? You are going to get multiple copies of the fragment of a particular DNA. Okay, say suppose for the same COVID that I told you, that scientist, he has identified the virulence factor, he has identified the gene for the antibody. Okay, say suppose he is not having multiple copies, he will not be able to give it to many people. So any piece of gene or any piece of DNA, you want multiple copies, then you are going to go for this PCR. Okay, so it was invented by Carey Mullis in 1983, and he mainly used Taq polymerase enzyme from Thermus aquaticus bacterium, which was isolated from that. So this enzyme, what is the specialty of this enzyme is the Taq polymerase enzyme, it can withstand high temperatures around 95 degrees Celsius. It can withstand. Okay. Now, 3 important steps. You will all know these steps. You will study for your board exams, all that. The temperatures are very, 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 very important. You know what is a primer? It is a small oligonucleotide sequences. Okay, it is a small oligonucleotide sequence that is going to contain a anticodon. Okay, you need not say it is an anticodon, a small oligonucleotide sequence that is known as enough. Okay, it is going to consist of 3 particular steps: denaturation, annealing, and then extension. So denaturation, you are going to unwind the 2 strands of DNA, that is called as denaturation. So what is the temperature? 94 to 98 degrees Celsius, but mostly many textbooks says it is around 95 degrees Celsius. But remember this range, 90s is for denaturation, followed by annealing. So the process of introduction of the primer into the unwinded strands is called as annealing. Usually happens at 50 to 68 degrees Celsius. Again, most textbook prefers around 54 degrees Celsius. We can take it in some 50s. So 90s for denaturation, 50s for annealing. Elongation, that is this to the primer, there are going to be various nucleotide sequences being added. So if you can see, the green color one which is present is nothing but your primer, and the yellow ones are the nucleotides. So all these are going to be added. It takes place at 72 degrees Celsius. So you have denaturation, annealing, and then extension. So there are 3 steps, and this is temperature that we must know. So I'll be uploading more of such videos. Please comment whether it is useful, you want me to improve in any of the ways, and I will try to include any MCQs as much as possible. I'll be leaving my contact number also with this video when I upload. in the comment section. You can check. So if you have any questions, any doubts, you can kindly let me know. Thank you for listening.